Welcome, everybody. Thanks for coming to this webinar from the New South Wales Pharmaceutical Science Group. This is going to be on vaccine technologies and an update of where we're up to. So this is just a very quick introduction from me, Scott Colborne. I'm the chair of the Farm Group in New South Wales, and happy to have two speakers today. We've got Dr. Dai Hayward and we've got Professor Pall Thordarson. Dai's going to speak first. He's in Japan actually at the moment, but based in the UK. And firstly, a big thank you to ATA Scientific, who basically organised Dai to present today. So a big thank you to them. So we are in a webinar today. I know most people are used to webinars, but just as a little bit of an intro here, just for today, as an attendee, microphones and videos will be disabled for everybody. If you do have any questions, we do want a Q&A. We do want that interaction. So please use the chat functionality. You'll see it does vary from platform to platform, but you should see the little speech bubble icon somewhere in the middle of the screen at the top, depending on what you're using. If you click on that, you get show conversation. It will bring it up. If you got a little red dot, it shows that there's something that's been posted, and it should appear on the right hand side. You get the meeting chat. Feel free to be providing information on that, asking questions, all that kind of stuff, and I will ask those questions to the presenters as we go on. And the last thing is the session will be recorded and will be available at a later date, so we'll provide that later on once we've just made sure the recording's okay and all the rest of it. So that's it. As enough of an intro from me. I'm going to hand over to Dai to start talking first, and then we'll have Pal afterwards. So over to you, Dai.
Well, good afternoon, everybody, and thank you very much for inviting me. It's a pleasure to be here. Let me introduce myself while I'm sharing my screen. I'm Dai Hayward, and I am the CEO of Micropore Technologies. And I hope everybody can see that screen.
Yeah, we can see that. That's perfect, Dai.
Good. Okay, so what I'd like to do is to talk to you about how we innovated, the journey of innovation that has led us into the RNA vaccine field, and how we've taken it from, if you like, the very smallest sample all the way through to manufacturing at a billion dose of vaccines scale. But first, where do we start? We start with, if I can get this thing to move. There we go. We start our provenance. The company has been around 20 years, and our provenance is in making things like PLGA microspheres. And you can see there that we have something that is really very, very nicely monodisperse, targeted around about 25 microns with a very narrow coefficient of variation. So a couple of years ago, as the COVID pandemic was raging, I had an idea, and I wondered if our technology would be adaptable to be able to use for making RNA vaccines rather than microspheres. In other words, could we make the jump down to the nanoscale? So I enlisted the help of Professor Yvonne Perry at the University of Strathclyde in Glasgow. I threw her one of our devices and I said, will this make LNPs, lipid nanoparticles? To which the answer a couple of days later came back, yes, it does. At which point we then had a problem and a challenge on our hands. So we did a lot of research, and we looked at the way that things were going in terms of how LNP development was working. Because what we were seeking to do was to identify a robust manufacturing route. And I have a history in manufacturing stuff rather than at the lab bench developing it. So I always look for something that's quite robust. Anyway, here are some of the parameters that are at play when anybody starts off thinking about a vaccine and its development. You've got the various different nanoparticle types. Because we're using RNA mostly, there's a bit of an alphabet soup about those, and that's not a full list by any stretch of the imagination. There are a variety of excipients that you can play with. And then not least at all, is the IP litigation risk. And anybody who's following this field will know that at the moment, pretty much everybody's suing everybody else. And it's all going to be a lot of fun and a lot of money earned by lawyers, and probably quite a lot of heat and not a lot of light. But needless to say, you've got an awful lot of variables there in order to come up with a formulation that you can take through into manufacturing. How do you approach that? Especially when I've heard somebody say, this is probably the most complex drug delivery system mankind has ever made, ever attempted. Well, the formation sounds really simple. You take your lipid mixture in an organic solvent, you add it to your aqueous buffer containing your RNA, and at its very simplest, you can do that in a beaker, and you can get LNPs. Not very even LNPs, and most certainly not reproducible, something that a regulator would not like at all. But forming them is the first step. So you've got the mixing to form the LNPs. You then dilute, because the ethanol that you would typically use as the lipid solvent means that the LNPs are unstable. So you need to dilute the ethanol down to stabilize and trap the RNA. And then you've got to do something. Oh, I've got dilution twice. Apologies. You've then got to do a buffer exchange and a reconcentration. And then finally, a sterile filtration and fill finish. And there you are at the bottom, you've got some LNPs. But there's a lot to go wrong in that. Different vesicles, different formulations will give different sizes. If you process badly, you can get the wrong sort of material with a very wide size distribution, which gets caught on the sterile filter, and therefore you lose yield, or indeed it's too big completely, and then you lose the lot. So the manufacturing process is integral to making sure that you get the right therapeutic outcome. And that manufacturing process requires a high degree of control. So let's look at where we start. There are two long-standing methods of making LNPs. On the left-hand side, you've got microfluidics, where you use a plastic chip and a very narrow channel and quite a high pressure pump. And on the right-hand side, you've got brute force and ignorance, effectively, where you put the two streams together under high pressure and you allow them to mix in that way. Now, the two processes both have limitations. Microfluidics, anybody who's involved in the field knows that they do not scale. And there are other issues as well, like the plastic chip. If you want to move away from ethanol and be a bit more creative about the sort of materials you use, you probably need to change solvents. And that leads you straight into interesting territory as far as the regulator is concerned. Because what that means is you have leachables and extractables to talk about with the regulator. On the other hand, the T-mixer, very nice when you've got a nice plateau, as you can see there. But once you move away from that with varying flow, it kind of falls off the edge of a cliff. I'm not saying these things. These are said by people who operate them, who we've spoken to on many an occasion. So we set about trying to remedy this. So we set out with a development objective to overcome those limitations and to develop a single process through all of the clinical development stages. And I'd like to take you through some of those stages as we go. So at the very first experiment, you've got a grey box there which talks about in the ideal world, you want to start small, preferably less than 0.5 ml of material, because RNA is eye-wateringly expensive, especially at the beginning. You need about 0.2 mils to be able to get the first reasonable data of physical and in vitro data out of it. You want to have something that's repeatable, and so on down the list. Our response was to develop what we call the AXF Mini, which is the little steel thing you can see there. But we've embodied it in what we call the Pathfinder. And the Pathfinder is a process-controlled, software-controlled design of experiment, high-throughput screening device. And it will give you 200 microliters per sample into a 96 well plate, and it'll fill that 96 well plate in less than a minute. And everything is reusable, so there are no consumables at all. So that kind of ticks the box as far as the starting point is concerned. The research objective was achieved with that. We then move on. So we now start to scale up, because now we're beginning to move towards preclinical and controlled data and dossier production and all of that. But you still need to keep low volume, you still want high repeatability, and you still want various other things like, low cost of operation, low waste, and so on. Funnily enough, the Mini does the same thing. Because it's a continuous process, you can go from that 200 microliters in the first example I showed with the Pathfinder Pro, the Pathfinder 20, and by the time you've added some more bells and whistles to the Pathfinder, you can go up to a 250 mil sample. The graph you can see there, if I were to enlarge it, you would actually see it's an N of 3. There are 3 different colors there, honest, but this was done at 100 mils a minute. We got a particle size for this particular formulation of 70 and a very nice narrow PDI of 0.13. So again, tick, we're moving on. This is looking good. Now at this stage, we're beginning to go into the clinic. And at this time, we want rather more material. We probably want a couple of 100 mils or maybe a little more. Absolute requirement here is reproducibility. Absolute requirement here is GMP. And here we go. The same device, the AXF Mini, will do exactly the same thing and get you that material through the Pathfinder Pro 1000, where in fact you can go up to about 5 liters. Still, however, being able to do 200 microliters for your very first experiment. So in one instrument, you can go from your very first experiment all the way through into phase 1 clinical trials for any reasonable sized disease population that you want to study. It's all very predictable. It's all very modeled. We've got excellent CFD relationship with the empirical data. So we know that this is a controllable process. And both this and the previous device, the Pro 250, are both certified to GMP and 21 CFR Part 11. So all good stuff. And then when it comes to the final version, we can go larger, we can go for the Mini, or we can go for one that's 10 times as long, and that's the AXF1. So instead, this time of having a capacity of 20 litres an hour, we go up to 200 litres an hour. And all of the repeatability, all of the scalability is fine. We've got minimum downtime here by comparison with other systems. And that is because there are no moving parts here at all. And we can integrate dilution and go on into, when we finish the development, an integrated continuous TFF. But you can still do TFF as a batch process if you want to. So, all good so far. How does it work? Well, this is the little schematic that we use. And you can see we've got a membrane at the left-hand end showing some pores. They don't actually, in real terms, come to the end of the device in the way they're shown there, but it's for illustration purposes. The RNA goes straight down the middle. The lipids go around the membrane and infuse through the pores, mixing instantly with the RNA giving you the giving you your LNP. Note the pressure. 2 to 5 bar is all we operate at. Very low. By comparison with both microfluidics and T-mixers, where you're talking of 30, 50, 60 bar. And I want you to pay attention for a moment to the little graph on the left. I've already shown you the one on the right, which is the T-mixer, what happens if you change flow rate. For ours, if you change flow rate, you get a very predictable behavior. And I'll return to that later. So, in summary, we've got 3 devices. We've got the AXF Mini, AXF1, and then we multiply up in a modular way, and you can see there the AXF4. With those flow rates, RNA concentrations and doses per dose, you can see the total production in terms of millions of doses of a pandemic vaccine or even a cancer vaccine that you can produce. And the AXF4, just to put it in the context, is about half the size of a laptop. So it's a tiny, tiny footprint by comparison with the wall of equipment that you may have seen that graces the Pfizer manufacturing facility in Kalamazoo, courtesy of CNN. So all looking good. And then we go to scale up. And here is a unit that uses the AXF1, which is sitting there, and then followed by another similar beast, which is capable of making 900 million doses. And the second one gives you inline dilution. So you've now got 2 stages of your process, which are harmonized and working together. And the box in which it sits is about 1.5 meters high, about 1.5 meters long, and about 0.5 meter wide. So very, very small. And the hold-up volume on all of that is still tiny by comparison with the T-mixer arrangement. That's a bit of a dog's breakfast if you've ever seen the CNN videos. Okay, all very good. Does it work? Well, there's an example of a formulation, very standard formulation. If you're into this, then this is nothing unusual. The NP ratio of 6:1 is absolutely normal. The total flow rate, the flow rate ratio rather of 3:1 is pretty standard. This uses poly-A instead of RNA for the expense reasons that I've already mentioned. We produce this at 100 mils a minute, and lo and behold, we got a 70 nanometer with a PDI of 0.13, and we did it 3 times, which is great, really good, very pleased. But this is the really interesting bit, at least as far as the FDA is concerned, because the graph on the right shows the curve that I mentioned earlier. And it relates to 2 formulations. The 2 curves are 2 different formulations based on the inherent chemistry of what you're doing. So they'll be different in, for example, PEG, the PEG lipid concentration. The PDI is still quite tight, but what you've got here is something that says, let's say on the bottom one, if I want to produce a 70 nanometer material, it's about there, which means around about 90 to 100 mils per minute, which is what I referred to earlier. So that was that formulation. Obviously, this formulation is inherently larger, down to 100 nanometers and up to about 160, which for most people is too large, except I've heard some arguments to say actually larger is okay. But the illustration that you can see on the left-hand side is something done in Australia, where the Mini, the AXF Mini, was taken at 2 rates, one at 20 mils a minute, and it produced a size of 107 nanometers with a 0.15 size distribution. Increase that size to 200 mils a minute, and the size comes down to 55, and the PDI gets to be 0.06, remarkably tight. Now that relates to this particular formulation, and obviously, you know, as financial advisors say, not everything does exactly the same. But what we've got is that predictable scalability. And the really interesting thing about it is that the FDA like this because it means you can start to think about introducing process controls that are automated. You can introduce PAT, process analytic technologies. So if you can put an online analyzer onto your process, then you can use the output from that when you have a deviation in size. You can use the output from the analysis, and you can feed that back to the pumps so that you can then control the pumps to give you the right size again. The FDA are currently installing this in their new continuous manufacturing facility, and we're looking forward to seeing what kind of results they get on that. Do we have good encapsulation? Yes, we do. This was Yvonne Perry at Strathclyde who came up with these numbers, 97, 98% very typically. I don't know quite what went wrong with the 91%, but certainly at the flow rates that we're looking at, then 90, 97, 98 is fine. And in fact, actually one customer and Yvonne have reported a few occasions where they've had 100%, which frankly I don't believe, but boy does it sound good. So then to the biology. This is Yvonne's work, not ours. We haven't got the biology. We've got the high encapsulation efficiency that I mentioned. Because it's a gentle process, we have no RNA degradation. Do we have a dose response curve? Yes. Do we have strong cell expression? Yes, we do. Do we have the mice that show where it's localized? Yes, we do. Does it produce an immune response in this particular instance? Yes, it does, based on ovalbumin. So it all works absolutely fine. And just to make sure that this is not a fluke, my grandson at the age of 14 came to do a day's work experience for me a little while ago, about a year and a half ago now, and he mastered the operation of this, the AXF Mini, very, very quickly. And in the afternoon, he was asked to make some LNPs by himself, and he did. And there is a video on our website if you choose to look. Oh, it's here as well. We might as well let it run. It's 30 seconds. We've advanced a long way since then. And this guy is not a scientist in any shape or form. He far prefers his languages. So I've lost him to the cause, but never mind.
There's still time, Dai. There's still time. He might.
Yeah, I know. I'd like to hope so. I really would like to hope so. So that was the waste, and now switching over to the good sample. And that was quite a lot of waste then. As I say, now with the Pathfinder, the waste sample is down to 300 microliters, and then the rest of the 96 well plate is 200 microliters for each well of good sample. So you get one waste sample, everything else is good for as many samples as you wish to run, anywhere between a few and 90 odd. So that's it really. Did we achieve what we set out to do? Yes, we did, and more in reality, because we were surprised about the ability to do the online control, and I can't spell scales. It's fully GMP. There is no plastic here, no single-use plastic of any sort, so there's no leachables conversation. The really interesting thing is you can go to that nearly 100 million doses using exactly the same core instrument. So when it comes to a conversation about moving through the development phases with your regulator, you do not have a conversation about scale up, because the only conversation is we run it longer. So there's no validation required. The data is the same because the conditions, the operating conditions, once you've settled on them, remain the same throughout the scale. And by the way, it does transfer from the size 1 to the 10 times size readily. So that's me. Thank you very much. Any questions?
We don't have any yet, Dai. That was wonderful. Thank you for that. The scaling thing, so that's the like qualifications and things wouldn't be required basically, because you're not really changing anything when you scale up. That's what you were saying at the end there, wasn't it? So
yeah,
is that
that's
right?
the beauty.
Yeah,
that's the beauty of continuous processing. That's why the FDA is really keen on continuous processing, because you're not changing the vessel parameters, the size, the stirring rate, or anything of that sort that can give you variability in your product. What you are doing is just simply using the conditions to run for longer. So if you're manufacturing your 200 microliter samples, you're doing so in less than 1 minute. But if you continue with the chosen settings, you can continue for 24 hours. And of course, because it's a continuous process, it's settled down very quickly, and the same product comes out at the end of the 24 hours as at the beginning.
Yeah, cool. Look, I see James has got his hand up. I'll turn his microphone on, see if this works okay. Hopefully this will work for you, James. Are you there, James? You need to unmute yourself if that's the case. I can unmute you for you if you want. No, I can't. Or maybe we can't. If not, if you put it in the chat, James, because we're in a webinar, sometimes some of the microphone and thing don't turn on. But otherwise, you could put your question in the chat if that's okay.
Stunned you all into silence, have we?
They're very shy, obviously. I see a couple of people have got their hands up. I'm just not sure if I can turn your microphone on. So if you could put your questions in the chat, that would be much appreciated. I'll ask the questions to Dai and then to Pally moving forward. While we're waiting for that, Pally, are you on the line there?
I am.
Okay, great, great.
And
great.
But if I'm allowed to, maybe I'll ask a question, Dai.
Of course.
It was a wonderful presentation, Dai. Could you go into a little bit of details, because in this space there's been a lot of also kind of discussion about the IP around the instrument itself and the licensing conditions if you go to GMP or manufacturing.
The approach here, I mean, again, different instruments, different technologies use or employ different business models. Our model is fundamentally that we start by saying we'll help anybody through their journey all the way through, including to get qualified for GMP. And in fact, we will be commissioning a couple of units early next year. And then we want to stay with people to make sure that the equipment stays up and running. So we look to put in place a long-term service agreement, bit like anybody having an HPLC serviced annually. We would expect to do the same to make sure the equipment is top-notch, bit like having your car serviced, really. You know, every now and again you change the tires. Well, you know, as part of that, we would expect to refurbish elements or maybe change the membrane or something. But this is a stainless steel membrane, and we started developing the technology for microspheres back in 2016. And in the lab, we're still using some of the original membranes from 7 years ago now. So if I wanted to develop a razor and razor blade business, I failed miserably.
Thank you.
No worries. What we'll do is I'll probably just, James asked his question first, so if we just answer that one, and then we'll let you do your talk, Pally, and then we'll come back to the questions afterwards. And if we don't have time, maybe we can fill them in on the chat anyway, gentlemen, if that's okay. So what are the formulation factors affecting the release of the core mRNA? Was James's question, Dai?
Oh, I think that's probably rather more in Polly's bailiwick than mine. I'm a mere engineer. I mean, there are things that will make a difference in the formulation, like the types of lipids you're using, and the way that you remove the ethanol in the process. The process of TFF is quite a rough one, so that can impose damage on the product. But in terms of understanding the chemistry, I'm not going to profess to be any kind of expert here at all. There are a lot of prevailing and countervailing issues around choosing the chemistry.
I can add to that a little bit. The very short answer is we don't actually really know. If I was being frank, I mean, it's still very much a trial and error science, and it comes down to one number, and that number is 1%. That is the quantity of mRNA that gets into the cell, that actually gets into the cytosol. 99% is still lost in the endosomes. Exactly why it's at least 1% and why it's slightly more or less with certain lipid formulation, I don't think anyone can answer that yet. And that's the harsh truth.
We don't exactly have 50 years of experience yet, though, do we, to be honest?
We have a few years, but look, the journey of nanomaterials through the cell is one that we in the nanomedicine field have been studying for 10, 20 years. And
right, okay.
I feel like we're just still at the, I wouldn't say quite the Stone Age, but we just entered the Bronze or Iron Age with understanding how nanoparticles in general traffic through cells or through the body.
Yeah, cool.
And that's the reality.
Fair enough. And on that note, you can talk more about how it's all progressing in Australia and in New South Wales, if you like. And then, yeah, we'll bring you back on, Dai, afterwards, if that's all right, and we'll do some questions. Yes, and maybe you make some comments in the chat for those questions, if you don't mind, in the background, if you feel like you can. If not, we'll chat online.
Well, thank you for the invitation, and Dai, thank you so much for bringing us to speed on some of the. I mean, you have really an example of frontier technology in what is actually probably the most important step in the whole manufacturing process. And thank you, David and Scott and everyone for the invite. I gave a talk a year ago, and I thought what I'll do, I will have sort of two parts to this talk. One is I will be a little bit of repeat, but also update on what's happening in Australia. And then I want to talk about one particular project that we are doing here. I think will be of interest to many of you. And of course, the talk here in more general will have a little bit of New South Wales focus, but I will try to cover what's going on in Australia. So let me see if you're seeing my screen.
Just loading up now. Just give it a second. Yeah, perfect.
All right. So, and I just want to make sure I could put the laser pointer on. Yeah. So, thank you again for the invitation. So, this is my mud map of what the RNA ecosystem in Australia looked before COVID, and there was maybe 2 companies doing something in that space. And our CSL had some R&D activities, and then PYS Antisense in Perth was doing some R&D. In terms of research support, this is what the map looked like. There was nothing, all right? There was nothing specific to RNA pre-COVID. And as I outlined in my previous talk, and I'd like to sort of start off with, if you want to, like we've done here the last 3 years, try to establish a new kind of manufacturing ecosystem, it's good to look at that sort of developmental framework, the technology readiness level framework. We mapped that on biopharma. Essentially, preclinical is sort of stage 3 to 5 or 2 to 5, and then 6 to 8 is sort of the phase 1 to phase 3, and 9 or 10 is your prototype. And in this, like so many other fields, I actually came across this first in solar cell research. There's a valley of death once you've done your initial eureka moment. in preclinical work. It's expensive, it's slow, no one wants to give you money for it. In Australia, once we get to clinical trials, the problem has been in many sectors that there's no local manufacturing, which means then often that the IP increase that happens in that phase doesn't actually realize itself in Australia. Look, I came into this from a research point of view. I was interested in RNA. I've been working on a little bit pre-COVID, particularly connections with Archer-Fox. Then mid-2020, this group of scientists kind of self-assembled. We didn't really know each other beforehand, and we, in a roundabout way, we initially self-assembled to help Colin Powton at MIPS, who was then already developing an mRNA vaccine. Back then, he wasn't getting too much support, so the Australian RNA Production Consortium that we formed essentially started lobbying for him. And this snowballed in the end, and the Victorian government finally started supporting his work, and they started, ITT did the first formulation for clinical trials in late 21, and the clinical trials phase 1 started in Melbourne last year. And I was just met Colin last week, and I think he said they are wrapping up the phase 1 trial now, so we will hear very soon about how that went. This group was expanded in 21 to include people from Queensland and South Australia and New Zealand. And yeah, it's been a nice venue for sort of in amongst the state competition, which I will be frank about has been happening. We tried to, through this avenue, also bring a little bit of Team Australia approach to this whole thing. And the federal government now is going through a consultation process about a national RNA plan. So just to refresh some of your memory here, the federal government did get involved in 21 with the approach to market and simultaneously negotiation with Moderna. The long and the short of that process was that they decided to do a deal with Moderna and they decided to build the factory in Melbourne. That is an end-to-end facility that can make up to 100 million doses a year. But obviously, we'll focus on Moderna products. It's not a contract manufacturing facility. It's a facility for Moderna, but it's a deal with the federal government, I believe, includes commitment to then make the vaccine here locally, particularly in if there was another emergency. But it would have to be probably a vaccine that Moderna has IP to. Few notable development outside the 3 big states where most of the action has been. It's actually been also there's a really good research center been formed at ANU, and I was there last week for the formal opening, although it started sort of in 21, the San Dalgano Center. And in Adelaide, Bridgewest bought up what used to be the Hospira site, and then Pfizer, and converted into Biosina, and they moved into plasmid production already at GMP level, and they are expanding into mRNA and lipid nanoparticle formulation as part of their services. In Perth, as I said, PYC was there pre-COVID. It's based on antisense oligonucleotide therapeutics developed by Sue Fletcher and other, but they moved into now morpholina oligos with cell-penetrating peptides that treat eye diseases, and they just did their first IND filing for that work. Syngenis is a small custom synthesis company that is also kind of a Murdoch spin-off company. And then Bridgewest has bought up what used to be the Pfizer fill and finish facility in Perth, creating Novacina. So now Bridgewest has both the Biosina facility in Adelaide and the Novacina in Perth. And in fact, this investment company also owns a number of companies in New Zealand and in Korea. Now, Queensland, more things going on there. The University of Queensland with Therapeutic Innovation in Australia increased from the base facility there in 21, and we kind of partner up with them now through TIA. Southern RNA is a sister company to a Canadian company. They've been working on R&D in Brisbane since mid-21. ProNATA is a startup company out of Griffith University who specializes in LNP formulations. And then Sanofi, that's the big deal. The Queensland government with the UQ and Griffith did a $280 million deal with Sanofi to set up a large R&D facility in Brisbane. And as far as I understand, though, not necessarily about GMP manufacturing, it's more about R&D and a big R&D headquarters for Sanofi there in Brisbane. And then Acura Bio was formed in 23 out of what used to be Luina Bio on the Gold Coast. And Cytivia and Acura Bio are partnering to offering, again, GMP production of plasmids, CDMO, and probably moving also into mRNA in due time. Now, Victoria, as I say, mRNA Victoria, the strategy in Victoria is controlled by mRNA Victoria, which is an offshoot of the government. They played a key role in helping Colin with that mRNA vaccine program. They obviously had a huge role in getting Moderna to Melbourne, working with the federal government. And that's in the Australian Singleton car park, as I say, it's outside Monash University in Clydeson. They're building the facility now. There's more than a hole in the ground. They're told it's starting to come out of the ground. It might be ready end of 24, I heard last week. And then they also done a deal with BioNTech about setting up an R&D facility, possibly small-scale GMP, in what's called a BioNTainer near La Trobe University, mainly focusing on cancer research, and again, probably more for products that BioNTech has IP on. We're waiting to hear more about this one. I want to highlight CSL as well because it is Australia's leading company in gene therapeutics, and I think that's actually one of the key future areas for RNA. Seqirus was formed, if I got it right, when CSL bought out the Novartis influenza vaccine business. And Novartis at that point, 7 years ago, was actually working on cell-amplifying mRNA, so-called siRNAs, flu vaccines, and CSL has been continuing this development. Might got the details slightly wrong, I hope not, but that's essentially what happened. CSL was involved in the HAM process, but then later on said they wouldn't want to try to make a homegrown COVID vaccine, but wanted to focus on the flu. And they're doing most of the work so far offshore. And they actually got some really interesting, I think now clinical results actually for their flu vaccine, and also done a very large IP deal with Arcturus in the US, who own some of the key IP in this space. There's also a small startup in Melbourne, Messenger Bio out of Melbourne Uni, which offers custom mRNA synthesis services for research. Now, so my story through this here in New South Wales is convoluted, but mainly building on my work in nanomedicine and also on the fact we brought someone here to UNSWn to set up a research group that does RNA synthesis. And kind of all these things converge to me initiating discussions with the chief scientist office pre-COVID about we should go into RNA manufacturing. And then long and short is that eventually the politicians thought that was a good idea. And what decision was made here 2 years ago was that it would be a collaboration between the universities and the state government. And what we essentially developed with the state government was this business plan, and this is my drawing of it, it's not an official, but in essence, the universities work together, and we already formed a pilot network in 21. Each university may do whatever it wants, like forming an institute, like we've done here and at ANU. But the key investment from the state government is in two initiatives. One is to support a CDMO GMP pilot facility, which is now going to be operated by Myeloid. I'll come back to in a second. And then also a 10-year R&D plan with $120 million investment. And the idea is that this will catalyze work from here and elsewhere that eventually would lead to clinical trials and manufacturing here, and obviously both bringing in international clients, creating startup companies and whatnot. The facility that the RNA GMP pilot facility will be built in Macquarie Uni, provided by, so the university provided the land, excellent location, just in that sort of pharma HQ of Sydney. Through a tender process, Myeloid Therapeutics was picked as the operator. They do have Charles River as a secondary partner as well. And they will hopefully be operating in 25 and offer CDMO services for both mRNA and synthetic RNAs and the lipid nanoparticle formulation of these. There's indirectly a link here to, so the factory will be owned by the state government, but leased out to Myeloid. And state governments also invested in a GMP viral vector facility at Westmead. So even though they're two separate operations, there's an indirect link because in certain formulation, you might want to use both in certain gene therapeutics. Not in vaccines, obviously, but in those. And just briefly about the institute we set up here, we have three kind of research themes: biology, medicine, and chemistry. And then we have this accelerator facility It's really trying to bridge the gap between the bench and then the GMP facility that Myologis is building, and in fact, possibly other GMP services around the country. By giving access people to pre-pilot scale, pre-GMP, and I would say actually we can, as we realize now, we can actually offer probably at least APMA approved GMP products in certain situations. Now, and again, so it's all about all the different modalities of RNA, not just mRNA. But at UNSWn, we have a lot of expertise in nanomedicine and nanoparticles. And as you heard from Dai's talk, that's kind of where a lot of the magic happens. And really, RNA therapeutics, it's actually all about the assembled nanoparticle. It really, that's the active ingredient. Before we have that, we don't really have anything. Now, we are therefore, as I say, providing materials predominantly for preclinical work, and we're also helping to train the workforce and work with related centers around the world. And within our institute, we have particular human health focus on COVID, and both mRNA vaccines and antiviral based on sRNAs. Very interested in oncology, as over a decade of work here on sRNA treatment of cancer, and there's a lot of work here around Sydney in personalized medicine. And we are also interested in rare and genetic disorders, treat those with RNA therapeutics, and we have a pediatrician involved that does that already here at Randwick Hospital. And there's a lot of action in the CRISPR space heading that way as well. And I think in the future also we realize that the pathway to understanding and possibly curing some neurological disorders and neurological degenerative diseases is probably through RNA. So what we call the RNA accelerator is the facility we built, so we can do the almost end-to-end production, as we say. We can make the starting material to make mRNA, which is the plasmid. I will come back to this again. We can make then the mRNA. We can purify it, characterize it. We can do the synthetic RNAs too, any different ways you want. Typically, synthetic RNA are up to 80 to 100 nucleobases, whereas mRNA a few hundred up to 10,000 nucleobases. And then we can formulate these into lipid nanoparticles and then characterize these. So we started in October 21. That's when the first people were hired. We didn't really get anything going until March 22, and by November 22, Josh Peterson and his team was the manager, had already achieved ISO 9001 certification. And we moved into brand new labs, 200 square meters, 15 staff. We have a clean room. We've done already over 100 batches of various RNA therapeutics for preclinical work. And we can offer people help with synthetic RNAs, with plasmids, with mRNA, self-amplifying mRNAs up to grams, and lipid nanoparticle formulations. And this number will probably go bigger as we are looking into solutions like the one Dai was showing you. And so we're very big on the being sort of, most of us actually like come from a chemistry background, so very big on quality control, USP guidelines, and we got some of the equipment that TGA uses, and I'm particularly proud of the mass spec facilities we have. And as I say, we achieved 9001 certification for our local production, and we can access also enzymes and other things through the recombinant product facility and the analytics of the genomics facility. Now, so that's sort of part one of my talk. Except to say, so this is the Australian landscape now. It's getting very busy. And I mentioned all of these except over in New Zealand. There's also South Pacific Sarah, which is moved into plasmid and wants to move into mRNA. I think I mentioned everyone else here. And in terms of the research R&D system, we've got the base facility, our institute, the Shang Delgado Center and ANU. We've got an innovation hub between Monash and Melbourne Uni. And as I said, some of that, I don't know really actually how much of this Queensland deal goes to the universities, but I'm hoping they will get some money out of it. The university sector here is getting $12 million a year over the next 10 years through a partnership with the government. MRI Victoria is supporting their universities generously as well. We know the South Australian government is doing the same. We don't know exactly how much. And there's something called the New Zealand RNA Technology Platform Program over the next 10 years with $70 million New Zealand dollars involved. Right, so that's sort of where we are at. I will go to the second part, which I think some of you might find quite interesting. And that's the story about Australia's first mRNA animal vaccine. Now, the picture here is of the Elizabeth MacArthur Agriculture Institute, which is out at Menangle, just south of Sydney. And it's an absolutely fascinating facility. It's one of the top agriculture research stations, I would say, in the world. It's the second largest biosecurity lab in Australia. And this team here, Peter, Paul, Jim, Valeria, have been working on this project now for a while, and it's funded by New South Wales Department of Primary Industries. And the foundation of their work is really a partnership between the New South Wales DPI, Tipo Biotech in Boston, and the Canadian Food Inspection Agency in Winnipeg, which happens to have some of the viruses of interest here. Now, we then became involved as a partner to assist with the scale-up. And obviously, down the line, we expect the myeloid facility and others to get involved in this as they come online. And there's also a vaccine and RNA design center at UTS that's been heavily involved, and Gary Myers has been heavily involved in this work. And what DPI is really wanting to drive is international collaborations for specific animal diseases of interest here locally. So what they've done, they have an R&D plan, so identify the antigens, test mice for immunity and so on, and the chemistry and manufacturing to GMP level, targeting animal safety, and the regulatory submissions to APVMA. The first of the here in this project is border disease virus. It's called BDV. It's in sheep, and it causes a lot of basically pregnant ewes can lose their lambs very easily due to this virus. So it's really destructive in terms of the economics for sheep farmers. And it's a single-strand virus. And it's endemic already, but getting worse. And this is kind of a good and a bad thing because it's endemic. There's no biosecurity issues in studying it locally, but it's a real need for the local farming community to try to stop the spread of this disease. So that's the first one this consortium is working on. We use a dendrimer formulation that TPA has developed. And building on this work, we will move on to lumpy skin disease, which is mainly in cattle and is and now in Indonesia, a major biosecurity threat for Australia, and also foot and mouth disease, which again, mostly in cattle and is raging through Indonesia at the moment as well. So I'm not a biologist, I don't know exactly what I'm looking at here, but I'm just showing here some of the data that Jim has been sharing with us. So they've done mouse experiments for the BDV, done the challenge. That looks all good. And then a few weeks ago, they vaccinated 30 sheep, and then they challenged these sheep. And what's not in this sort of data format, but I can tell you, is that it works. I'm told by Jim that those sheep are showing both protection and good antibody titers after injection with that BTV vaccine, which was made by TPA in Boston on a small scale. And then what we are working on here at UNSWn is to scale that up. So that's our next step, to scale it up. And I just want to use that sort of as a segue just to recap a little bit how these things are made. Being a chemist, maybe one way to approach the problem is to actually think about it almost from the opposite end. It's like a retrosynthesis. What is mRNA? mRNA is made from DNA in our cells, and then it codes for the protein. So that's what mRNA does in our own cells. So what we want here, in case of vaccine, is to make an antigen. We want the cells to make an antigen. But rather, but we want to inject the mRNA somehow directly into the cell. And we deliver it, therefore, into the cells. We're not editing or changing the DNA at all. This is why these processes are not really anything to do with gene regulatory technologies. We're not changing the DNA. We're just putting mRNA into the cell temporarily, and it won't last very long. And we do this with a nanoparticle that gets taken up by endocytosis. And as I said, only a very small portion of the RNA actually gets into the ribosome. But it's enough to get translated and form the antigen of interest. And it will be gone after a couple of days, if not further, shorter time. Now, how do we get to that nanoparticle system? Well, so basically, you think about drug substance in mRNA vaccine, it's really just this assembled nanoparticle. If you don't assemble this into a nanoparticle, you don't have a vaccine. The mRNA, if you inject it directly, will last a few minutes in the blood and it won't do anything. And in fact, just make you probably very itchy. The lipid nanoparticles not only will not do anything if you inject them, they actually don't have the same structure quite if you package them without the mRNA. So it's the assembled nanoparticle really, which is a drug substance, and it has mRNA cargo, it has active lipids, as we call them, like what could be dendrimers, and then it has helper lipids like cholesterol and POPC. Plus, normally you would then formulate this in a buffer with for injection and some cryoprotectants like some sugars. That's it. That is what you're making. Now, there are many steps on the way to that, especially to make the mRNA. You start by identifying the antigen, then you design your RNA sequence. You actually then generate complementary linear DNA, which is usually done in E. coli, then linearized and isolated. And then everything after that is completely cell-free. And you just take the DNA template, an enzyme, a few hours, the nucleotides, you get your mRNA, you purify it, like very similar to protein purification. And then those things are mixed with the lipids to do the actual formulation of the drug substance, which is the assembled nanoparticle. So just to take this in slightly more detail, so PNA, this is very similar to what people do in protein production, except well, we're not after the protein, we just grow the bacteria and then lyse the cell, isolate all the DNA, chop it up with enzymes. There are quite a few purification steps involved, and end up with a sterile, completely sterile sample of a DNA plasmid. That then is put into an enzymatic reactor with some enzymes and nucleotides, and that's what generates about 100, 200 copies of the mRNA. You put in an enzyme, digests all the DNA away, and then there's a couple of more purification steps to get rid of the salts and so on, chromatography, buffer exchange, and you get your ready to formulate mRNA. Now, this process, someone mentioned in chat, it's actually very expensive simply because some of the nucleotides that are used are very expensive, especially this so-called capping unit. And that's probably, this one is hard to make. It's a 10-step synthesis, so that's why it is expensive. But a lot of this has to do with IP. So there's strong IP on this one, so they charge it through the nose for that one. And if you use modified nucleobases, many people do to reduce the immunogenicity, they're also very expensive. So actually a lot of the cost comes from these two IP-protected starting materials. And then, as I said, the assembly step is the main one, and you saw this from Dai. You can do this in many different ways. There's a laminar flow or T-cell, T-flow or whatever, or the technology that I was showing you. But you end up with your, well, in our case, for the animal project, we're using dendrimer nanoparticle, but essentially they have the same shape. It's just the active lipid is a proprietary dendrimer. And from a polymer chemistry point of view, these particles are somewhat like a core shell. So the active lipid and the mRNA are mostly on the inside, and the helper lipid, the POPC, and the cholesterol and the PEG form the outer shell. But it's a little bit more complicated than that. But to a first approximation, it is a bit like a core shell type structure. And typically, yeah, 50 to 100 nanometer in size. So that's the summary. Really, this is the main step. Everything else is just basically you're mixing together lipids or dendrimers or whatever it is. And the mRNA, there's quite a few steps to get to the mRNA, but it essentially starts with making a plasmid. And the plasmid is the starting material to make the mRNA. And obviously a lot of other raw materials involved here. And then, but it's really to make the particle, which is the active substance. You have one, pure mRNA, and two, the lipid mixture, and that's what makes the nanoparticle. So for us, this is the key step in the QC and regulatory type monitoring. So I just want to thank you for your attention and thank my wonderful staff, and happy to take some questions.
Yeah, thanks. Thanks, Pally. That's great. We are at 5:30, but if you guys are happy to stay on for a little bit, we'll do some questions if people can stay on, and we'll go through that. So what I'll do, are you still there, Dai?
I am.
Okay, great. So I know, thanks, Dai, you've answered a couple of questions in the background. That's wonderful. Thank you.
Looks like I've answered them in the wrong order. Sorry.
That's all right. That doesn't matter. There was one about, forgive me if you've already looked at this. It talked about, does lipid polymorphism impact on stability of LNPs? Looking at DSPC, it may potentially have an impact. So did you answer that one, or?
I did.
Okay.
I must confess I'm not the expert, as I said
Right.
earlier,
Okay.
but I do know that DSPC is one of the commoner lipids used.
Right. Okay.
Yeah, I haven't heard any problems with that. So
No.
I
Yeah,
in
I
fact,
agree.
yeah. So the lipid nanoparticle isn't so much the problem, actually. If you have the right device to make it, like Dai's device or something else, the immunogenicity of the particle does, though, probably depend a little bit on your formulation, as in which lipids are involved. But
Yes.
the nanostructure, I mean, they are so large in a sense. They're 50 to 100 nanometers. I don't think, these factors kick in compared to some of the other factors we have to worry about.
Right.
Probably more important is the particle is good enough, shall I say, to shield all the RNA away from the body. Because if there's some RNA sticking out, that's when your immune system will go crazy before you get to your target.
Right. Fair enough. Well, everybody's been a bit shy, I think, now with any more questions, so maybe they've asked them all already. We'll give it just a second. If not, we'll wrap up, and just let me just give it a second to see if anybody comes through. Otherwise,
I mean,
unless you want to elaborate on one of the others.
Well, I mean, as I said earlier,
welcome.
The expensive thing is actually mostly to do with the raw materials, and it's mainly the two used in the mRNA. It's the pseudouridine and the capinucleotides. I did calculations, and I've seen other people where if you made 1 billion doses, they are 44% of the cost, including factory, people, the whole shebang.
Yep.
And it's mainly because of IP.
The rest of the process is relatively inexpensive compared to more traditional techniques as well, or. So is it still more expensive than others because of those ingredients? I understand why they got to be expensive. I'm sure they didn't. I didn't knock them up in 5 minutes, did they? So
I mean, my argument is not. Like, I mean, like if you were to try to basically make X milligrams of protein in your body, it will be much cheaper to do it this way than making a protein in CHO cells. Okay. You see what I mean?
Mm. Yeah, fair enough.
But I want to do the proper comparison one day. Actually, Dai, you might know this. Micropore technology differs from piezoelectric manufacturing. I wasn't quite sure about.
I've not heard of piezo.
No, I haven't actually either. So,
but not in this context anyway. So I'm a little unsure about the basis for that question.
Now, David, you asked us about FDA should consider this one batch, as in one like 24-hour run or something. David, I'm not. I know that what the TJ told me is that when they were getting the pallets from Pfizer and Moderna, is that they would batch test the batches that came from the overseas here in Canberra. Yeah, but that is because, as John Skerritt explained, this is getting injected into millions of healthy people. They would not do that if this was a cancer therapeutics.
Right. Yeah. I mean, in fundamental terms, the FDA and other regulators, but the FDA particularly, has been driving for at least the last 30 odd years the idea of continuous manufacturing, and they have been incentivizing at the API level that if you know your process and can control it, they'll cut you a bit of slack on allowing process changes in a way that traditionally they don't. And I think that is applying now to other areas of continuous manufacturing, including vaccine manufacturing.
Right. Right.
So there is still a conversation to be had about what is the definition of a batch. Of that, there is no doubt. However, they're becoming a little more lenient about engaging in that conversation compared with where they were before.
And that's
David,
what
Is
No,
it
go on,
great
sorry.
yet? No, it's not. Is it, is it perfect yet? No, it's not. You still hear, from a manufacturing point of view, you still hear, a lot of wailing and gnashing of teeth.
Yeah, fair enough.
and
I think
like,
David made a comment. Sorry, Pally. David made a comment there about sampling regimes of once every 24 hours kind of thing and that kind of thing. So
yeah,
speaks to the same thing. Sorry, Pally, carry on.
No, I was going to say it was a very interesting comment John Skerritt made in a meeting here a few months ago, that if I understood him correctly, the FDA is now leaning towards really, and also Scott McNeil, who has worked for the NIH on toxicology of nanoparticles, that really what they consider is the assembled nanoparticle, because there is no point worrying about, they say, the toxicity of the polymers in the nanoparticle, because it is not a nanoparticle at that point. So its immunogenicity is going to be completely different when it's not assembled versus.
Yep.
So there's really no point anymore to worry so much about the components, because the particle itself is the active ingredient. If it's not a particle, it's not an active ingredient. And likewise, if you make a particle so badly there's naked RNA around, then, well, that's going to cause problem on one hand, but it's also going to disappear in a few minutes in the blood. So so yeah, the focus is all now on the batch testing of that particle and making sure that the particle is doing its job, and that includes actually testing whether it's making the right protein. So part of the TGA process for human vaccines too, that they actually test in vitro on cells if it makes the right protein. That's more important.
And that gives you a different issue, because actually some of this, in terms of the biology, as you go through from cell to mouse to, and then up to human through different mammals, there is not a straightforward read across in the biology results. And that causes a regulatory challenge for sure.
Now, Roger Reed mentions the proprietary technologies are the opportunities. You betcha. there's a arms race of people trying to come up with alternative lipids and chemistries to avoid these really expensive ones. That being said, I've encouraged some of my medical friends to say, well, do you really want to fight that battle, or just develop your platform based on something you can license and then just sleep better afterwards? The other approach I've seen some of the Boston-based companies do is that rather than using the most expensive, say, capping nucleotide, they go for an older technology that works. It's not as good in terms of efficiency, but it works, it's safe, and obviously much cheaper. So if they show the efficacy in other ways, it's a win-win, right? So, and I think you've seen this with the lipids discussion too. Some people just go back to something simpler.
Absolutely.
Simply.
There are some very complicated lipids out there, and there are all sorts of strategies being deployed to functionalize or decorate the lipids as well. And that's an interesting challenge. Part of that is to avoid IP. Part of that is also to try to enhance the tissue targeting, which itself is a major challenge that we've not referred to in this. Is
that a different talk altogether, is it? Tissue targeting.
Oh yeah, no, I mean absolutely, and that's yes. Actually stated.
Just noting down the next ones I'm going to get people to talk about.
Well, I was going to say that, actually, I said this is our stated #1 challenge of our institute, is actually targeting, and many people think, they might be wrong, that the best way to target some of the tissues is not through lipid nanoparticle, but through polymer nanoparticles.
Yes, right.
And then possibly by, again, functionalizing them with either peptides or sugars or even antibodies. And yeah, so that's going to be a whole new game. Conventional lipid nanoparticles work very well as for vaccine delivery, because they seem to reach the lymph node system very well. They also go very easily into the liver.
Yeah.
When it comes to other tissues, it's not quite as easy.
So what we might do is, we got one last question just come through. If we can, we'll go with that one, and we'll wrap up, gents, if that's okay. So with ongoing RNA modifications and novel LNP formulations, how do you anticipate changes in regulatory pathway and policies going forward? Sounds like a simple question.
You want to try that one, Dai, first?
Thank
right,
you,
and that's
Polly.
the end of the webinar. No, sorry.
I mean, I think this is one of those things where This whole field has suddenly burgeoned because we've done the biggest clinical trial in history, with the COVID vaccine. It arose out of two things that came together effectively. The identification of a particular kind of lipid, the ionizable lipid in Vancouver, and the ability to make RNA stable with the pseudouridine that Polly referred to. I think, as Polly said, we're very much in the foothills of this. There's an awful lot yet to understand, some of which is how do these things perform? How can we get a bigger load into the cell? How can we do better than 1%? What are the issues about targeting, and the regulations and policies that will come out of that will respond to the scientific advances, I think are still very much in their infancy. It was all done in a real hurry first time round because we needed to make the world safe. Now the FDA and others have gone back to their rather more ponderous way of doing things, and things are taking time in a way that they didn't with warp speed and all of the political initiatives that drove it forward. So I think we're going to see a rather more considered approach, and some of the questions that maybe were not answered or not fully addressed in the COVID vaccines, I think are going to come back to the fore. So some of the issues around side effects, some of the off-target effects and things like that are going to be issues to be debated and resolved. I don't know how you think, Polly.
I partly agree. I will say the one issue I feel we're still dealing with is also almost like a information fog. And because some of these issues already have been resolved, it's just that 80–90% of people working in space don't know that. And I mean, in some ways, like with the lipid nanoparticle, I think there's no doubt the only reason why it was approved so quickly was that there was already the Onprato lipid nanoparticle formulation approved by the FDA in 2018. So there was already an approved FDA drug for rare genetic disorder based on almost identical lipid nanoparticles 2-3 years on the market. So that was actually a huge reason why the FDA and EMI, I think, had that comfort with what BioNTech and Moderna are doing. Plus, they had done a trial up to phase 1 or 2 on the Zika virus 2-3 years earlier. So it wasn't just like it happened the first time. And this is what I feel sometimes a bit frustrated. You have to blame things over and over again. For instance, here in Australia, at least what's here from the TGA, the OTTR ruled some time ago. I mean, RNA is not OTTR. You don't have to notify about OTTR with working RNA unless you do the actual gene editing, like with CRISPR. So you don't have to do it during manufacturing. The mRNA vaccines are not OTTR yet. We continuously have to try to explain to people it's not subject to OTTR because you're not editing your genes, right? But part of the reason is that there was also this crazy crowd out there that actually said mRNA vaccines are editing your genes. So there's almost this sort of information fog that is sort of here and there. And it's amazing that, the knowledge that already is within the FDA and TGA, for instance, on this matter hasn't maybe permeated as much out as I would have liked to see it doing. But yeah, I spent a lot of my time the last 2 years, even, I mean, I had to educate politicians and starting with explaining what RNA stands for, right? So.
Yep, I know that one.
So, no, but I agree with you. There's also now, especially once we move into all the tissues, all the type of modalities, all the type of therapeutics, in some ways, the human vaccines is the extreme, right? Because as John Waskerrit was trying to explain to me, you are then subjecting healthy individuals. It's very different from a cancer patient that is very sick, right? Your bar is much higher with a healthy individual, right? But the flip side, though, is that it's only once or twice a year, right? Whereas if you're doing a continuous RNA therapy, you might need injection every week. So these are the matters we haven't sort of quite seen coming through, right?
Yeah, and if you're doing it if you're injecting every week, then you've got to watch out for the vaccine creating its own immune response.
Exactly. Yeah.
From the.
No, I mean, well, it is not a vaccine, but it starts to act like a vaccine. That's the problem.
Yeah.
And I mean, in the vaccine space too, there's no doubt about the fact that the lipid nanoparticle itself is somewhat acting like an adjuvant.
Yes.
But again, we don't really understand exactly how. But I mean, it's one reason why it works, right?
Well, thank you, gentlemen. That's been wonderful. I've kept you another 16 minutes over, so I apologise for that. But
That's
thank
okay.
you. I think everybody's
That's
enjoyed
okay.
those questions. So thank you again, and thanks everybody for attending. I'll put a quick link in the chat to where you can find past events and also where the future events for RACI are if you want to come along. Don't be a stranger. Bit far to you to come to a face-to-face Dai, unfortunately, but we'll see. Never know. But yeah, thank you again, gentlemen. That was wonderful. So enjoy the rest of your evening, and see you soon. Thank you.
Thank you.